The Biostatistics Shared Resource Facility is one of the core resources of the Markey Cancer Center that is available to all Cancer Center members. The mission of the BSRF is to foster collaborative interactions among members of the Markey Cancer Center in the provision of scientific and statistical support in all aspects of cancer research. The BSRF collaborated with the Markey Cancer Center's small animal imaging facility to provide guidance regarding experimental design, data collection, and statistical analysis of xenograft and transgenic mouse models of cancer. The goal is to ensure that studies are designed properly in order to adequately address research hypotheses and collect and store high-quality data. The following basic elements should be considered in designing your animal studies. These first 4 items are primarily the responsibility of the investigator, but early discussions with biostatisticians can help you refine your research goals and design. First, you need to clearly define your research objective and be able to translate this into testable statistical hypotheses. What is the null hypothesis of your study, which is usually the hypothesis of no difference, and what is the alternative hypothesis of your study? The alternative hypothesis is your research goal and is the complement of your null hypothesis. The second element is the primary study endpoint. What endpoint will best capture your research objective? Additional questions to ask include: when should you measure this endpoint? Do you measure this endpoint over time? When are the appropriate time points to measure your endpoint? What are the additional data that you're going to collect, such as secondary as well as exploratory endpoints that will be measured at time of animal sacrifice? The third element is: what are the experimental factors that you want to evaluate in the study? This will help you determine the number of groups, the types of treated and control groups that you will use in the experiment. This will also help the biostatistician decide what is the most appropriate study design for your experiment. Fourth, what is the appropriate animal model to use, say for a tumor growth versus a metastatic experiment? Do you need to perform a small pilot study to fine-tune your animal protocol? Some of the protocol parameters that you need to evaluate include duration of tumor development for your particular mouse model, tumor take rate, variability in time to tumor development for transgenic models, number of cells that will be injected in order to have adequate signal intensity. The rest of the elements listed here are statistical considerations that can be addressed in collaboration with the biostatistician. The information from the first 4 elements that are already mentioned earlier will facilitate discussion and development of these statistical considerations. The study design is based on your primary study endpoint, the experimental factors of interest, and your research hypotheses. There are several options that can be used to design your experiment as listed here. The next element is the sample size, or the number of animals per group. This is a critical component that needs to be planned ahead of time. In addition to providing justification required by the Institutional Animal Care and Use Committee, this ensures that you are able to adequately address your research hypothesis with sufficient statistical power. The main element in calculating sample size and statistical power is what is called effect size. This is a measure that takes into account the hypothesized amount of difference in your study endpoint between study groups and the amount of variation in the study endpoint. This information has to come from the investigator, not the statistician, and can be based on pilot or preliminary data or published studies that can closely approximate specific aspects of your proposed study. It is important to ID your animals within each group. especially if they are being monitored over time. The last two items, randomization and blinding, are strategies to minimize bias in your study design and study conduct that might affect the resulting data. Randomization allows control of any extraneous factors that might confound treatment effect. Some of these factors include animal weight and tumor size at baseline. Blinding is another mechanism to reduce bias in treatment comparisons. Ideally, the experiment should be done blind to diminish the possibility of subjective evaluation of outcome. For example, the main investigator may need collaborators or co-workers who will be responsible for measuring, collecting study outcome that is blinded to the treatment group assignment. We recommend that data collected in the small animal imaging facility use the database system developed through LabKey. This will facilitate accurate, clean, and complete data collection and data entry for timely statistical analysis. Please refer to the data repository LabKey link in this website for use of this database system. Contact information for the BSRF is markey.biostat@uky.edu.